अस्सलाम वालेकुम डियर स्टूडेंट। आज के इस लेक्चर में हम डिस्कस करेंगे माइटोसिस। इन प्रीवियस लेक्चर वी हैव डिस्कस्ड अबाउट इंटरफेस एंड द phases of interface, which was G1 phase, S phase and G2 phase. Mitosis actually contains two division, karyokinesis and cytokinesis. Actually, uh, karyon mean nucleus and kinesis mean division. When we talk about karyokinesis, it means we are talking about division of nucleus, and when we talk about cytokinesis, it means we are talking about division of cytoplasm. Second thing, why mitosis is important and how mitosis takes place. Actually, mitosis takes place in somatic cells, and somatic cells are the cells of a body of organisms. And when a zygote is formed by the fusion of egg and sperm, that zygote contain a single cell. But an organism which is a multicellular needs to form many cells of the body. In order to make many cells from a single cell, mitosis needs to take place so that a single cell can be converted into multi cells. So in mitosis, the number of chromosomes remains same as in parent. For example, if the parent chromosome contain 46 chromosome, then the daughter cell which is formed from that parent cell will also contain 46 chromosome. So mitosis takes place only in eukaryotes, and in prokaryotes, mitosis does not take place because in prokaryotes there is no membrane bounded organelle. So they also not contain nucleus. So prokaryote divide the cell by a special division which is called binary fission, but it is not mitosis or cytokinesis. So mitosis actually reorganize DNA in the cell. Prior to mitosis, as we have discussed in interphase, DNA needs to duplicate. So before mitosis, the DNA will be duplicated in S phase of interphase, and during mitosis, the two copies are reorganized, repacked into two sets of chromosomes, and divided to opposite end of the cell. And in most organisms, mitosis is immediately followed by cytokinesis. It means when the karyokinesis takes place, immediately cytokinesis will also be occurred. And in mother cell, and as I already discussed, in mother cell and daughter cell, the chromosome number will be same. Here you can see on the screen, it is a nucleus, and inside the nucleus there is a thread-like structures which is called chromatin, and this structure develop before cell division. And as you know, the DNA and protein make chromosomes. So when the chromatin will be condensed, this structure will be developed, and this is called chromosome. And in one cell, there are 23 pair of chromosomes are present, and in whole body, for example, if there are millions of cells and multi cells of that particular organism, all the cells will carry the same number of chromosomes, and all the cell carry same genetic information. So in mitosis there are different phases. The first one is prophase, second is metaphase, then anaphase and telophase. And before telophase, cytokinesis will be takes place, so a cell can be divided into two separate cells. Here is a summary of whole mitosis, and we will discuss one by one what changes takes place in prophase and what changes takes place in metaphase, anaphase, telophase, and at the end how the single cell divided into two cells. The first one is prophase, and in prophase, chromatin is being organized into chromosomes, and it is present inside the nucleus, and microtubules which develop from the centriole. And centrioles is a non-membrane bonded organelle which is present inside the cytoplasm, will be moved towards the opposite pole, and four centrioles are present in one cell. and two centriole at the same time is called centrosome. Before during early prophase, inside the nucleus, there is a thread-like structures visible, which is called chromatin, and with the passage of time, that thread-like structure start to condense and form a chromosome. So in prometaphase, here is a condensed structure, as you can see here, which is in the form of chromosome, and centriole develop the microtubules, which start to bind on the kinetochore region. Here is a kinetochore protein, which is present at the centromere of a chromosome, and the microtubules start to bind at that region. In metaphase, all the chromosomes set and align at the equator and at the center of the cell. So, in one centriole, from one centriole, microtubule arise and bind with the kinetochore, and on the other side, the same centriole, which is present on the opposite side, originate a microtubule, and it also bind on the same chromosome, kinetochore of the same chromosome. Then in anaphase, the chromatid start separated because one chromosome contain two sister chromatid. So the sister chromatid start separated and start moving towards opposite pole, and this is the identification that a cell is in anaphase. And in telophase, cell chromatin arrive at opposite end of the cell and begin to unfold into loose coil of the chromatin. And always keep in mind the telophase is actually the reverse of prophase. Now, new nuclear envelope began to form around the chromatin to create two nuclei, and spindle fibers disappear into cytoplasm. The cell membrane began to cleave in preparation of cytokinesis, and in cytokinesis, in plant and animal, the cytokinesis is different. In animal, because there is no cell wall, so the simple division will be takes place in the form of cleavage. Cleavage is a special type of division which actually divide the cell into two. And in plants, because there is a cell wall present, so it's hard to divide a cell with cell wall into two cells. So a special organelle which is present in the plant is a Golgi apparatus, and that Golgi apparatus start to form a plate which is called phragmoplast. So phragmoplast help in cytokinesis in plant, and cleavage and actin and myosin protein help to divide the cell into two cell, and in cytokinesis in animal cell. Here are some animations that easily describe about mitosis, and I will play one of them. Uh, just wait a minute, I will play, and you can easily understand how the mitosis can be takes place.